tell me about tomorrow's protocol with Ace2 that you're doing with the kids.
Oh, well, first of all, Ace2 is a very important gene for humanity in the last 5 years, because it makes the protein that sit on the surface of your cell, and that receptor, we call it, what recognize COVID-19. You know, it can be floating around, but until it hit that protein, it cannot be recognized. And once that it get pulled inside the cell, and that's when you're infected. But tomorrow they are going to do a 32-step DNA isolation protocol, trying to collect the whole DNA genome. And there's a kit that you can literally drool. You know, I kid them around. I said, "Oh, come on, you need to drool into this tube." You know, they're basically collecting saliva, and the cheek cell in your mouth are in the saliva. That's where the cells are, and that's where they're gonna go in there and they're gonna break open all the cells. You know, all these 32 steps, and they are gonna somehow pull out just the DNA nice and clean, isolated, so that we can do experiment after that. Because we need the, you know, and they're gonna isolate their own DNA genome, and then we can use a technique called PCR, polymerase chain reaction, just like any of the DNA lab around here. We're gonna use that protocol to go in there to their whole genome, and we're gonna pull out just one gene out of it. Once that they have completed this whole unit, they're gonna learn number of labs, they're gonna learn how to do sequence analysis, they're gonna learn how do we develop drugs and vaccine against COVID. I'm gonna walk them down the street to Alexion, because we're at Career High, so it's like 4 blocks of walk, like 3 or 4 blocks. The kids still complain, but you know, we walk in there, and they are gonna meet real scientists working on making discovery from some of the basic techniques that they use in the lab.